for another allergy library video. So I look a little off, but I've been out and they had the windows down and they're like in the wind and all. So yeah, today we're on Pathophysiology, and it is a disordered physiological process associated with disease or injury. Now, it was first recorded in between 1950 and 1955. And pathophysiology and physiopathology are convergence of pathology with physiology, where pathology is a medical discipline that describes conditions typically observed during the disease state, whereas physiology is a biological discipline that describes process or mechanisms operating within an organism. Pathology describes the abnormal or undesirable condition, whereas pathophysiology seeks to explain the physiological processes or mechanisms where such a condition develops and progresses. Pathophysiology can also mean that functional change is associated with or resulting from diseases or injury. Another definition is for functional changes that accompany like a particular disease. So yeah. Now the term of pathophysiology comes from the ancient Greek word of pathos and physiologia. In like the 19th century, in like the reductionism in Germany in the 1830s, Germany's mother led the establishment of physiology research autonomous from medical research, and in 1843, the Berlin Physical Society was founded in part to purge biology and medicine of vitalism, and in 1847, Hermann von Helmholtz, who joined the society 2 years prior in 1845, published a paper on the conservation of energy, which is highly influential to reduce physiology research foundation to the physical sciences. And in the late 1850s, German anatomical pathologist Rudolf Virchow, a former student of Müller, had direct focus in the cell, establishing cryptocytology as the focus of physiological research, while Julius Kleinheim pioneered experimental pathology in medical schools, scientific laboratories. And from there the germ theory began, and by 1863, motivated by Louis Pasteur, reported on fermentation to butyric acid, and a fellow Frenchman, Casimir Darwin, identified a microorganism as the crucial causal agent in the Castle disease of anthrax, but is routinely vanishing from blood left over, scientists inferring as it as like a mere byproduct of petrification. And in 1876, upon Ferdinand Kahn's report of a tiny spore stage of bacterial species, a fellow German, Robert Koch, isolated Darwin's bacteroids in pure culture, which was a pivotal step that would establish what would become bacteriology as a distinct discipline, and identified a spore stage that applied Jacob Henle's postulates and confirmed Darwin's conclusion. Which was a major feat of experimental pathology. Pasteur and colleagues followed up with ecological investigations confirming its role in the natural environment via spores in the soil. And also, as a septicemia, Darwin injected rabbits with a highly diluted tiny amounts of putrid blood, which can duplicate duplicated the disease, and use the term fermented petrification, but it was unclear whether these refer to, as did Pasteur's term, fermented to a microorganism or something like that, as it did for many things, to a chemical. And in 1878, publication Etiology of Traumatic Infective Diseases, unlike any of the previous work been done, where in 80 pages noted by a historian was able to show that in a manner practically conclusive that a number of diseases differing clinically, anatomically, and etiology can be produced experimentally or by injection of putrid materials into animals. Used bacteriology and the new staining methods with aniline dyes to identify particular microorganisms. For each, the German theory of disease crystallized the concepts of cause, presumably identifiable by scientific investigation. And in scientific medicine, the American physician William Welch, trained in German pathology from 1876 to 1878, including under Cohenheim, and operated America's first scientific laboratory, a pathology laboratory, at Bellevue Hospital in New York City in 1878, and Welch's course drew enrollment from students at other medical schools, which responded by opening their own pathology laboratories, and once appointed by Daniel Coit Gilman, finally advised by John Shaw Billings, as founding dean of the medical school of the newly forming Johns Hopkins University. Dr. Gilmore, as its first president, was planning. Welch travelled again to Germany for training in Koch's bacteriology in 1883, and upon his return to America, he moved to Baltimore, eager to overhaul American medicine. When blending Vickers anatomical pathology, Cohenheim's experimental pathology, and bacteriology, Hopkins Medical School, led by the Four Horsemen, Welch, William Osler, Howard Kelly, and William Halstead, opened at last in 1893 as America's first medical school devoted to teaching German scientific medicine, so-called. It's like in the 20th century, biomedicine was developed, like the first biomedical industry, Pasteur Institute and Berlin Institute for Infectious Diseases. His first directors were Pasteur and Co., were founded in 1888 and 1891 respectively, with America's first biomedical institute, Rockefeller's Institute for Medical Research, which was founded in 1901, with Welch nicknamed the Dean of American Medicine as its scientific director, who appointed his former Hopkins student, Simon Flexner, as director of pathology and bacteriology laboratories. And by way of World War I and World War II, Rockefeller Institutes became global leaders, a global leader in biomedical research. And from their molecular paradigm, 1918 paradigm triggered frenzied search for a cause, although most deaths were via local pneumonia, already attributed to pneumococcal invasion. In London, pathology with the Ministry of Health, Fred Griffith, in 1928, reported pneumococcal transformation from virulent to avirulent between antigenic types, like as it nearly switching species, challenging pneumonia-specific causation. In the laboratory of Rockefeller Institute, Oswald Avery, America's leading pneumococcal expert, was troubled by a report that they refused to attempt repetition. And when Avery was away from summer vacation, away on summer holidays, Martin Dawson, a British Canadian, convinced that anything from England must be correct, repeated Griffith's results. Then achieved transformation in Liberto to opening a precise investigation. And having returned, Avery kept a photo of Griffith in his office, in his desk, while researching. His researchers followed the trail, and in 1944, Avery, Colin MacLeod, and Macklin McCarthy reported the transformation of factors as DNA. Widely doubted amid estimations that something must act with it. At the time of Griffith's report, it was unrecognised that bacteria even had genes. And so I think a couple of years later, we found out that we had the structure of our DNA. And the first Mendelian genetics began in 1900, yet inheritance of Mendelian traits was localized to chromosomes by 1903. Post-chromosomal genetics, biochemistry emerged in the same decade, and in the 1940s, most scientists viewed cells as a stack of chemicals, a membrane containing loose molecules of chaotic motions, and the essential cell structures as chromosomes, which bacteria lack as such. And chromosomal DNA was presumed too simple, so genes were thought in chromosomal proteins. Yet in 1953, American biologist James Watson and British physicist Francis Crick and British chemist Rosalind Franklin inferred DNA's molecular structure, a double helix, which I just mentioned not that long ago, and conjectured to a spell code. In the 1960s, Crick helped crack a genetic code in DNA, thus establishing molecular genetics. In the late 1930s, the Rockefeller Foundation had spearheaded and funded molecular biology research programs, which seeks to fundamentally explain the organisms of life. Led largely by physicist Max Delbruck at Caltech and Vanderbilt University. Yet in the reality of organelles in cells was controversial and made unclear visualization with conventional light microscopy. Around 1940, largely via cancer research at the Rockefeller Institute, cell biology emerged as a new discipline, filling the vast gap between cytology and biochemistry by applying new technologies like ultracentrifuge and electron microscope to identify and deconstruct cell structures, function, and mechanisms. The two new scientists interlaced cell and molecular biology. And mindful of Griffith and Avery, Joshua Lederberg confirmed bacteria conjugation, reported decades earlier but controversial, and was awarded in 1958 a Nobel Prize in Physiology or Medicine at Spring Harbor Laboratory on Long Island, New York. Delbruck and Salvador Rivera led the macrophage group hosting Watson, discovering details of cell physiology by tracking changes to bacteria upon infection or with their viruses. And process transduction, Liedberg led the operations of a genetics department at Stanford University's Medical School and facilitated greater communication between biologist and chemical departments. And by the 1950s, disease mechanisms were developed in researches on rheumatic fever. A complication of streptococcal infections revealed it was medicated by the host's own immune response. Now, during investigation by pathologist Lewis Thomas, that led to identification of enzymes released by innate immune cell macrophages and that degrade host tissue. And later in the 1970s, as president of Memorial Sloan Kettering Cancer Center, Thomas collaborated with Liedberg, soon to become president of the Rockefeller University, to redirect the funding focus of the U.S. National Institute of Health towards basic research into the mechanisms operating during disease processes, which at the time medical scientists were all but wholly ignorant of, and too, as biologists were scarcely taking interest in disease mechanisms. Thomas became, for American basic researchers, a patron saint of sorts. And the uses of pathophysiology today is required to study for nearly all healthcare professional school programs, like whether it be medical, dental, physician assistant, occupational therapy, physical therapy, nurse practitioner, like radiation therapist, pharmacy, nursing, radiological scientist, chiropractic and paramedic programs in the United States, Canada and many other countries. And pathology of pathophysiology of Parkinson's disease is death of dopaminic neurons as a result of changes in biological activity in the brain with respect to Parkinson's disease, also known as PD. There are several proposed mechanisms for neuronal death in PD. However, not all of them are very well understood very well. And there's 5 proposed major mechanisms for neuronal death in Parkinson's disease, including protein aggregation in Lewy bodies, disruption of autophagy, changes in cell metabolism or mitochondrial function, neuron inflammation and blood-brain barrier (BBB), breakdown resulting in vascular leakness. The pathophysiology of heart failure is a reduction of the efficiency of heart muscles through damage or overloading, such as it can be caused by a wide number of conditions, including myocardial infarction, in which the heart muscle is starved of oxygen and subsequently dies, hypertension, which increases the force of concentration needed to pump blood, and amyloidosis, which is misfolded proteins or deposited in heart muscles, causing it to stiffen. And over time, these increases in workload will produce changes to the heart itself. And the pathophysiology of multiple sclerosis is that of an inflammatory demyelinating disease of the CNS with central nervous system, in which activated immune cells I forgot what I was up to. which activated immune cells invaded in the central nervous system and caused inflammation, neurodegradation, and tissue damage, and the underlying condition that produces this behaviour is not known currently. And the current research in neuropathology, neuroimmunology, neurobiology, and neuroimaging, together with clinical neurology, provides support for the notion that multiple sclerosis is not a single disease but rather a spectrum of diseases. The pathophysiology of hypertension is that of a chronic disease characterized by elevation of blood pressure. Hypertension can be classified by cause of either essential, also known as primary, or idiopathic or secondary. About 90 to 95% of hypertension is essential hypertension. Now, in pathophysiology, HIV and the AIDS involves acquisition of a virus. Thus, a virus replicates inside and kills T helper cells, what is known as T helper cells, no idea what they are, which are required for almost all adaptive immune responses. This is an initial period of influenza-like illness, and then a latent symptomatic response, a phase which the C4D gate count falls below 200 cells in every milliliters of blood. The HIV host has progressed to AIDS, a condition characterized by deficiency in cell-mediated immunity and resulting increased susceptibility to opportunistic infections and certain forms of cancer. And the pathopsychology of spiders biting is due to the effects of the venom of the spider, and the environmentation occurs whenever a spider injects venom into the skin, and not all spiders bites. It injects venom as like a dry bite, and the amount of venom injected can be based on the type of spider and the circumstances of the encounter with that spider. Now, the mechanical injury of a spider's bite is not a serious concern for us yet. It will when every spider literally kills us. And the last area, the pathophysiology of obesity involves many possible pathophysiology mechanisms involved in development and maintenance. The field of research had been almost unapproached until the leptin gene was discovered in 1994 by J.M. Friedman Laboratory. These investigators populated that leptin was a satiety factor of the O/mal. Mutations in the leptin gene results in the obese phenotype, opening the possibility of leptin therapy for human obesity. However, soon thereafter, J.F. Carr's laboratory could not detect any mutations in the leptin gene in humans with obesity, and on the contrary, leptin expression was increased, proposing the possibility of leptin resistance in human obesity. So yeah, this is the video today, so yeah, I'll see you all very soon, as I'm doing at least 2 more videos today, one being the news and another being my May Day bank holiday weekend and all that kind of thing. So yeah, I'll be back very soon for the both of them. So yeah, bye for now.